I will be talking about Gaucher disease. The GBA gene codes for glucocerebrosidase, otherwise known as GBA. This gene is found on chromosome 1q. Glucocerebrosidase is a 3-domain protein, with domain 1 consisting of a 3-stranded antiparallel beta sheet. This domain also has 2 cysteine residues that are completely necessary for the proper folding of the enzyme. And if you look to the arrows on the right, you'll be able to see in the top 3D structure where the N terminal is, as well in the bottom one there is a nice little loop that occurs there. Domain 2 consists of 2 closely associated beta sheets that resemble an immunoglobulin fold. If you look to the arrows on the right side, you'll be able to see where this fold is in the 3D structure as well as the drawing structure. Domain 3 consists of a TIM barrel, which contains the catalytic site of the enzyme. The arrow pointing at the upper figure is showing where the catalytic site is, whereas the lower one is simply pointing to the TIM barrel. 2 other points that are relevant to bring up are that domains 2 and 3 are connected by a flexible hinge. It's hard to see that in these figures, however. Another point is that domain 1 and 3 do tightly interact, which you can see better in the 3D structure than the drawing structure down below. Generally, glucocerebrosidase is a 497 amino acid protein, and it is 59.7 kilodaltons in weight. The 2 most important residues of the catalytic function of this enzyme is E235, which is an acid-base catalyst, and the E340 residue, which is a catalytic nucleophile. Glucocerebrosidase has a necessary cofactor named saposin C. Saposin C is the enzymatic activator of GBA and is completely necessary for its function. In fact, if saposin C is mutated, which in some cases it is, it results in Gaucher disease. Saposin C is 9.31 kilodaltons in weight, 80 amino acids in length, is a glycoprotein, and is a homotetramer. To activate glucocerebrosidase, saposin C must first embed itself in the defects of the membrane. This results in a thin membrane, which exposes glucocerebroside, the substrate of glucocerebrosidase. After this event, there are 2 proposed models of how saposin C may activate glucocerebrosidase. The first being the solubilizer model, where saposin C dissociates from the membrane, binding to glucocerebrosidase, activating it, and giving it its substrate glucocerebroside. In the second model, saposin C associates with the membrane and does not dissociate. Glucocerebrosidase comes to it, receives its substrate and is activated, and then leaves. In summary, saposin C mediates the contact of glucocerebrosidase with glucocerebroside, as well as induces a conformational change directly, which stimulates its enzymatic activity. Glucocerebrosidase is a part of the ceramide pathway, where glucocerebrosidase breaks down glucocerebroside, a component of cell walls, into ceramides. Ceramides play an important role in cell biology as they are precursors of sphingolipids, which are involved in the lipid membrane, as well as act as secondary messengers. Ceramides also are highly expressed in neurons and do many jobs associated with those cells. Interestingly, ceramides also are potent inducers of cell cycle arrest. This is interesting because glucocerebrosidase is inside of lysosomes, that is inside of macrophages, that break down old cells that have gone through apoptosis and or damaged cells. Glucocerebrosidase catalyzes the hydrolysis of glucocerebroside into glucose and ceramide. And this is the reaction. But in cells, it looks more like this, especially the lysosomes of macrophages, where glucocerebroside is constantly broken down by glucocerebrosidase. Glucocerebrosidase utilizes a 2-step mechanism to catalyze the hydrolysis of glucocerebroside into glucose and ceramide. In the first step, it utilizes acid-base catalysis as a nucleophilic carboxylate anion attacks the glucocerebroside molecule, relieving it of the ceramide molecule. The acid-base carboxylic acid then gives it its proton and releases the protonated ceramide out into the cell. The remaining sugar molecule is then covalently attached to the enzyme, one of the former carboxylate anions. From there, water comes in and attacks the bond between the enzyme and the sugar molecule, forming glucose. Another possible reaction is transglucosylation, where an alcohol comes in and does the same exact thing, resulting in not glucose, but another sugar product. Given this table from Michelin et al., the native Km of glucocerebrosidase is around 6.04 millimole per liter, and for Vmax, it is around 13.31 nanomole per H per milligram of protein. Glucocerebrosidase is uncompetitively inhibited by glucose. And since this is glucocerebrosace's natural product, this is feedback inhibition. This can be seen by the Lineweaver-Burke plot up top, where the lines are nearly parallel. This indicates uncompetitive inhibition, whereas gluconolactone, an artificial substrate, competitively inhibits GBA. This can be seen from the Lineweaver-Burke plot down below, where the lines intersect at the y-axis. To draw conclusions about the thermodynamics of glucocerebrosidase, we must look at an analogous enzyme. In this case, we are looking at TNBGLB, which is also a beta-glucosidase. Its substrate in this reaction is PNPG, an artificial substrate that is not very similar to glucocerebroside, so we must keep that in mind. We must also keep in mind that the delta S values and temperature values of this graph are pretty wonky compared to what we should expect. We should expect the delta S values to be positive and not super low, considering that it is breaking down one glucocerebroside into ceramide and glucose. Obviously, this is a different reaction, though, and the temperatures are not bodily. They are way beyond what life could sustain in the human body. Barring both of those differences, if we're still assuming that it is relatively similar to that of glucocerebrosidase, then we could assume at least that glucocerebrosidase has a positive delta G and is probably only pushed forward by a low product concentration and a high substrate concentration, especially since the mechanism that we saw does not involve any ATP, any cleavage of any CoA, or any loss of CO2. A mutation in the GBA gene is what causes Gaucher disease, and there are over 380 mutations that can cause Gaucher disease. However, the majority of the cases of this disease are caused by two mutations. For type 1, the N370S mutation is typically the culprit, and for Gaucher disease types 2 and or 3, the L444P mutation is usually the culprit. When there is a mutation in the GBA gene, this knocks out the activity or number of glucocerebrosidase. This in turn results in a deficiency in the breaking down of glucocerebroside, which increases the concentrations of glucocerebroside inside of certain cells. Comparative to the native values of Km and Vmax for glucocerebrosidase, those that are homozygous for Gaucher disease-causing mutations are significantly worse off as their Km and Vmax values are significantly altered. It can be seen that the Vmax values are significantly lowered, while the Km values are significantly increased, indicating a significantly lower enzymatic activity and significantly lower activity in the glucocerebrosidase enzyme. However, those that are heterozygous for one of these mutations are still significantly different, especially in the case of D409H. However, there is still the case of N370S, where its Km and Vmax values are nearly identical to that of the control group, which are those with the native Km and Vmax. A deficiency in glucocerebrosidase causes a buildup of glucocerebroside in the lysosomes of macrophages. In fact, the nucleus of those macrophages is pushed to the periphery of the cell by the accumulation of glucocerebroside. This type of macrophage that is affected by the accumulation of glucocerebroside is called a Gaucher cell, and they are particularly dangerous as they secrete lysosomal enzymes and inflammatory signals. Gaucher cells are said to have a wrinkled tissue paper appearance. Since Gaucher disease affects the lysosomes of macrophages, any tissue with high macrophage content will be affected. For instance, bone marrow, the spleen, the liver, and the lungs. However, Gaucher disease can also infiltrate other tissues and organs, which can then elicit its effects there as well. Gaucher disease is mainly seen in the Ashkenazi Jewish population, with 1 in every 800 births resulting in type 1 Gaucher disease. Type 2 and type 3, however, are not localized to this population. The high rates of Gaucher disease in the Ashkenazi Jewish population are due to high rates of the disease-causing mutations in the population. They are then passed down by an autosomal recessive means, and the mutations are retained in the population. In the normal population, 1 in every 40,000 to 1 in every 60,000 births result in Gaucher disease. Of the 3 types of Gaucher disease, type 1 is the most common, whereas type 2 accounts for less than 1%, and type 3 accounts for approximately 5%. Clinical presentation is one of the ways that Gaucher disease can be diagnosed, and one of the ways that the 3 types can be differentiated. Type 1 can be diagnosed at any point in one's life, and in some cases can leave those affected without any symptoms. However, other symptoms include hepatosplenomegaly, which is enlarged liver and/or spleen, eye movement disorders, and yellow spots in the eyes, and due to the effects on bones, anemia, which can cause bruising and fatigue. Type 2 is usually diagnosed in babies under 6 months of age and also shares the symptom hepatosplenomegaly with type 1. However, this type also includes movement problems such as muscle spasms and trouble swallowing, and usually results in severe brain damage, leading to the usual lifespan of these babies only being about 2 years of age. Type 3 is usually diagnosed within the first 10 years of life and is a combination of the previous two types, with similar bodily symptoms to type 1, but with neurological degradation similar to type 2 but with a slower onset. Those with type 3 may also struggle with seizures. There are two other ways to diagnose Gaucher disease. The first being genetic testing. This is to check for mutations in the GBA gene. This can inform you about yourself, but will also help to inform you about the chances of passing on Gaucher disease-causing mutations to your children. The second way is glucocerebrosidase leukocyte blood testing and/or enzymatic analysis of fibroblasts. Both of these ways test glucocerebrosidase activity and can be verified with serum levels of glucocerebroside, as those with Gaucher disease typically have elevated levels of 5 to 10-fold of glucocerebroside. In summary, you can use a variety of information to diagnose Gaucher disease, such as the age of onset, the clinical presentation, the ethnicity of the patient, genetic testing to test for mutation and to see which mutation is present, as well as testing for glucocerebrosidase activity and for the levels of glucocerebroside in the patient. Currently, there is no cure for Gaucher disease. However, there are two main treatment options. The first being enzyme replacement therapy, where the patient must inject themselves with recombinant glucocerebrosidase every 2 weeks, such as imiglucerase. This is to help bring down the levels of glucocerebroside in their system. The second being substrate reduction therapy, where the patient must orally administer inhibitors to the enzyme that makes glucocerebroside, or glucocerebroside synthase. This will also help with the accumulation of glucocerebroside in their system. Future treatment options may include gene therapy, where the GBA1 gene will be introduced into hematopoietic cells and then injected into the patient. That might help with producing active glucocerebrosidase. The second future treatment option would be introducing molecular chaperones into the patient. This would help with the production of functional glucocerebrosidase and may even help to restore the activity of mutant glucocerebrosidase. Currently, there is no treatment for type 2 Gaucher disease or the neurological symptoms of type 3. As for making connections, an easy one is looking at the supposed C mechanism of activation for glucocerebrosidase. As we saw earlier, it's a protein, and so a protein activating a protein is something similar to a zymogen, which is high yield and something that we hit on a lot in class. A second connection would be the active site catalysis, where we see an acid-base catalysis reaction happening here, as well as a covalent intermediate being formed, which is, interestingly, 2 of the 3 catalysis ways we saw this semester. Another connection would be the transition state intermediates, where you're able to see how these carboxylate anions are stabilizing the water and or alcohol groups that were taken off of the molecule, something that was introduced to me this semester. Another connection would be looking at the enzyme and understanding its folds, like the immunoglobulin fold, something that we didn't really learn but were introduced to, as well as the TIM-Burrel, something similar we didn't learn but were introduced to, as well as the catalytic residues and domains that we saw on this enzyme, something that my attendance of this class and learning in this class really helped to understand this enzyme for. As for metabolic pathways, the ceramide pathway wasn't covered a ton in this presentation, but it was still something relevant, and understanding the 3 big questions that we asked throughout the semester about the pathways and or enzymes, what does it look like, what does it do, and how is it regulated, played a big role in specifically glucocerebrosidase, but also played a role when looking briefly at the ceramide pathway. Now, when we're looking at the effects of an enzyme being deficient, that's something we dealt with throughout the semester, whether it be in questions or just trying to understand pathways such as gluconeogenesis or glycolysis or the TCA cycle better. But being able to understand that and look at the effects from that was something that helped me to understand how to research and how to go about understanding the deficiency of GBA in Gaucher disease. And lastly, feedback inhibition by glucose, where it is a natural product of the glucocerebrosidase reaction that it catalyzes, and coming back and inhibiting it is something that we see throughout the semester, and we have a nice uncompetitive Lineweaver-Burke plot to that was high yield and something that was involved in this presentation.
To reflect on learning about Gaucher disease and doing this project, honestly, I chose to study Gaucher disease on a whim. I heard people talking about them being mad at themselves for not choosing another disease that they had previously looked at, but choosing a different one. And I didn't want to deal with having conflicting thoughts about stuff. So I just chose one on the page, stuck with it, didn't waste any time deliberating. That way I had more time to focus on it. And I just stuck with it. I was like, I'm not going to switch. So it just so happened I chose one that people really liked. The most interesting thing I learned during this project wasn't a single point. It was seeing how everything was connected, especially seeing how, like, oh, this is connected to Parkinson's or this is connected to another pathway, which is really, really interesting. However, if I choose something specific, I think the formation of Gaucher cells is super interesting, and the buildup of glucocerebroside in the macrophage is really interesting. I've told at least 4 people in my family this already, and it's awesome. I think it's really, really cool. The most challenging thing and frustrating thing about this project was the amount of information was overwhelming. Unlike other projects where there's probably 4 papers total on them, this one had a lot of information, and so deciding what the narrative you wanted to tell was and not getting sucked away into a rabbit hole was difficult, but it was definitely a good way to know if you were telling the narrative you wanted to, if you got too much information. And lastly, I wish we had done mini versions of something like this for gluconeogenesis and glycolysis. Obviously not to the extent, but definitely learning about certain enzymes in this way was super helpful, because I know a lot about beta-glucosidase now, and it would have been really interesting to do something similar to this on a smaller scale for other enzymes or another pathway perhaps.